Hello everyone, my name is Santiago Castañeda Palacio. I am third semester medicine student and I will to talk you about evaluation of glutathione transferase, PI1 expression and gene promoter methylation in Moroccan patients with urothelial bladder cancer. Bladder cancer is part of genitourinary tract cancer. It affects older patients with a median age of 73 years. Is related with tobacco smoking, contaminated water on underdeveloped countries, and also is related with environment toxic exposure. Is more frequent in males than females. Glutathione transferase is a group of enzymes. Its function is catalyze the conjugation of reduced form of glutathione. On reaction, glutathione plus xenobiotic generates glutathione S conjugate. Is a cytosolic detoxifying enzyme. His main role is the maintaining of cell integrity. It inactivates a wide variety of electrophilic carcinogens or stress substances. Is an apoptotic regulator. And GSTP1 is related with esophageal cancer, renal cancer, lung cancer, breast cancer, and colorectal cancer. GSTP1 is also related with chemotherapy resistance. In this study, we aim to determine the expression status of GSTP1 in relation to this gene promoter methylation in Moroccan population of 30 bladder cancer patients and into non-cancerous bladder tissues used as controls. Materials and methods. 30 bladder biopsies were collected from urology department of the military hospital Instruction Mohammed. The samples were divided into groups. The first group for DNA extraction and the second group were stained with hematoxylin eosin. Immunohistochemistry is the determination of a specific molecule in a tissue. It's done through the interaction of an antigen and an antibody. Previously, the antibody was marked with a chromogenic substance. The cells are colored with the presence of marked substance. This technique was applied to GSTP1 determination in bladder biopsies. DNA extraction. This technique is realized to purification of DNA or RNA. First, the cells are extracted for the patient. After that is done analysis of the cells to liberate genetic material. After, the proteins were removed, and finally, DNA was concentrated. In this study, it's used to determination of GSTP1 gene sequence and bladder localization. PCR. This technique is used for DNA, RNA, and gene amplification. This technique involves in vitro enzymatic amplification of DNA, increasing its copies or increasing the copies of a specific gene sequence. In this study, it was used for amplification of GSTP1 gene sequence. GSTP1 promoters was amplified with primers for unmethylated and methylated alleles. Results. These were the GSTP1 amplified sequences. In this study, we can see that the range of age is from 42 to 78 years, with a median age of 65 years. It was evaluated the grade of invasion of cancer. It was classified in non-invasive tumors with 3 patients and invasive tumors that was classified into two groups: non-muscle invasive with 19 patients and muscle invasive with 8 patients. Histologically, the cancer was classified or evaluated with low grade or high grade. In control samples, the GSDP1 expression was strongly and moderately expressed in the other case. In bladder cancer samples, the expression of GSDP1 was moderate with 60% strong 23.13% and weak 14.33%. In one bladder cancer case, the GSDP was not expressed. The most important item of this table is the moderate classification based on immunoreactivity staging. We can see the patient with non-muscle invasive with 73.68% also classify to high-grade staging. MCF7 is a cell line used to determine epithelial cancer. Methylated as control and unmethylated alleles were amplified. In the 30 bladder cancer samples, the unmethylated alleles was detected. The low coloration of the samples of the patient 9 to patient 20 is related with all of these patients has a non-muscle invasive tumor grade, and all of them have high grade on histological classification. Pliesa Herzegovina et al. Its accumulation into the nucleus was ascribed to the overexpression BCL2 protein that has been implicated as the regulation of transport of GSDP1 through the nucleus pore complex. Negative. Martignano et al. During immunohistochemistry examination, dishomogeneous GSDP1 staining was observed in most of the tissues. This heterogeneous staining was also reported in the prostate. Positive. Sacristan et al. Found that GSDP1 promoter was methylated in 44.6% of cases, but the methylation decreased with cancer progression in classified Ta versus T1 stage and distinguished low-grade versus high-grade tumors. Positive. Conclusions. The expression of GSDP1 in bladder cancer tissue compares with normal tissue says that the presence of GSDP1 is variable in all tissues and it's not related with bladder cancer. And the integrated study of pathology is very important because it allows amplified the knowledge about this and also allows to know the different presentation of the pathology. And it was all. Thank you for seeing this video.